Hello everyone. In previous lecture we have discussed the cytoplasm. In today's lecture we will discuss ribosomes and its function. So this is the structure of ribosome. In ribosome mainly it is made up of two units. This is large unit and this is small unit. The total formation of protein synthesis depends on these two subunits, larger subunit and smaller subunit. And this is a amino acid chain, growing amino acid chain. Larger subunit accumulates the amino acids and collect it in a protein chain. So the binding of amino acids in a chain is known as polypeptide chain. So translation RNA is exists inside the large subunit and the mRNA means messenger RNA is exist inside the small unit. So basically this is a small structure of ribosome and the prime function of ribosome is to protein synthesis. We will discuss it in later in detail. Now we will discuss the ribosomes and its functions. So as we all know that ribosome are the important organelles in both prokaryotic and eukaryotic cells. And the ribosomes play a central role in protein synthesis. Despite their small size, they are essential for cellular function and survival. Without ribosomes, cell would be unable to produce protein, which are necessary for growth, repair, and regulation of biological activities. This lecture explores the structure, function, location, and significance of ribosomes, along with their role in protein synthesis and real life examples of their importance. First, we should know structure and composition of the ribosome. As I already stated the structure of ribosome. Ribosomes are small spherical organelles made up of two major components. Those are ribosomal RNA, which provides structural support and catalyze protein synthesis. Second is ribosomal proteins. These help in assemble and stabilize the ribosome's structure. Each ribosome consists of two subunits, as I already stated, large subunit and small subunit. In small subunit, it binds to messenger RNA and ensure correct translation. In large subunit, it joins amino acids together to form a protein chain. This is protein chain. Next is, before going in the next, we will take an example, ribosome as a factory. Suppose ribosome as a factory where the blueprints means mRNA means messenger RNA are used to assemble products means proteins. The small subunit reads the blueprints while the large subunit assemble the product. Next is role of ribosomes in protein synthesis. Protein synthesis is the primary function of ribosomes. It occurs in two main stages. First is transcription. In transcription, there is DNA to mRNA process. Before ribosome can produce proteins, the genetic code from DNA is transcribed into messenger RNA inside the nucleus. Second is translation. In this, mRNA to protein process occurs. Once mRNA reaches the ribosomes, translation begins. In translation begins, there is three steps. First is initiation. The ribosome attaches to mRNA at the start codon AUG. Second is elongation. Transfer RNA brings amino acids which are joined by peptide bonds to form a polypeptide chain. Third is termination. When the ribosome reaches a stop codon, the protein is released. For example, insulin production in the pancreas. The pancreas produces insulin, a hormone that regulates blood sugars level. The ribosome translates the insulin gene into an insulin protein, which is later released into the bloodstream. Third is types of ribosomes, means free versus bound ribosome. Ribosomes exist in two forms depending on their function and location in the cell. Free ribosomes means found floating free in the cytoplasm and synthesize proteins used within the cell. For example, enzymes production in muscle cells. Muscle cells need enzymes to break down glucose for energy. Free ribosomes synthesize these enzymes which help generate ATP, means energy, during exercise. Next is bound ribosomes. Bound ribosomes are attached to the rough endoplasmic reticulum RER and synthesize protein that are exported outside the cell or used in the mammal. For example, antibody production in human immune cells. White blood cells produce antibodies to fight infections. These proteins are synthesized by bound ribosomes and released into the bloodstream. Next is ribosomes in prokaryotes versus eukaryotic cells. Ribosomes are present in all the cells, but they differ in size and structure between prokaryotes and eukaryotes. In prokaryotic cells, prokaryotic ribosome 70S its main features. Location is free floating in cytoplasm and it is smaller, means 70S, found in bacteria and archaebacteria. In eukaryotic ribosome, this is larger one, 80S. It is attached to the RER, means rough endoplasmic reticulum, or in cytoplasm. It is found in plants, animals, fungi and protists. Now let us take an example. Bacterial ribosomes and antibiotics. Many antibiotics like tetracyclines and target bacterial ribosomes 70S to stop protein synthesis, killing the bacteria while leaving human 80S ribosomes unaffected. Next is ribosomal malfunction and diseases. Error in ribosomal function can lead to serious genetic disorders and diseases. First is ribosomopathies. Genetic mutation affecting ribosome production leading to disease. Diamond blackfan anemia, where red blood cells production is impaired. Cancer, we have already discussed it so many times in this. Uncontrolled protein production due to defective ribosome can contribute to tumor growth. For example, cancer and ribosomal dysfunction. Cancer cells often have hyperactive ribosomes, producing excess proteins that help them grow uncontrollably. Scientists are developing drugs to inhibit ribosomal activity in cancer cells. Next is importance of ribosomes in biotechnology and medicine. Ribosomes has led to breakthrough in medicine, biotechnology and genetics. In gene therapy, scientists use ribosome to synthesize therapeutic proteins such as missing enzymes in genetic disorder. Vaccine production, mRNA means messenger RNA vaccine like COVID-19 vaccine instruct ribosomes to create viral protein triggering an immune response. For example, mRNA vaccine and COVID-19. The Pfizer and Moderna COVID-19 vaccine use mRNA technology where ribosomes produce a harmless viral protein that teaches the immune system how to fight the virus. Next is evolutionary significance of ribosomes. Ribosomes are one of the oldest cellular structure present in all living organisms. Their universality suggests that they played a key role in the origin of life. So there is a theory about this, endosymbiotic theory. In this, mitochondria and chloroplast have their own ribosome similar to bacterial ribosomes, supporting the idea that they were once independent bacteria that merged with ancient cells. For example, ribosomes in mitochondria. Mitochondria have 70S ribosomes like bacteria, which support the theory that they have evolved from free living organisms. Next is the future of ribosome research. The scientists continue to study ribosomes to understand their potential in medicine, genetic engineering, and synthetic biology. Also in field of artificial ribosomes, researchers are working on creating synthetic ribosomes to produce custom designed proteins. Next is ribosome targeted cancer therapies. Drugs that inhibit faulty ribosomes could help stop tumor growth. For example, lab-ground ribosomes. For example, lab-ground ribosomes, scientists have successfully engineered ribosome in test tubes, opening the door for customized protein production. Lastly, we would like to say that ribosomes are indispensable in their role and one of the most important organelle in a cell, that is ensure essential proteins are made for growth, repair, and regulation. They function as molecular machines, translating genetic information into the proteins that drive life itself. From antibody production to muscle repair, ribosome influence nearly every biological processes. They are also vital in medicine and biotechnology, leading to advancements like mRNA vaccines, gene therapy, and cancer treatment. Without ribosomes, life as we know it would not exist. Thank you so much.